In this microbiology lecture, I will take a closer look at how we prepare specimens for staining, and then also some of the different types of stains that we can use in the microbiology laboratory. Bacterial cells are colorless, and if we wanted to try to look at them and try to observe any kind of detail about them, we wouldn't be able to see any type of detail if we didn't properly prepare them and stain them. In looking at using the light microscope, which is what we'll use in our lab, um, we can use two different types of fixes or two different types of preparations. The wet mount and then a fixed specimen. A wet mount is useful when we want to observe living organisms, um, typically. So we had a sample of pond water and we wanted to observe the organisms in that. We would simply take a drop of that pond water, put it on a slide, put a cover slip on it, and then we would have our preparation. Sometimes we could add stain to that. If we were, taking, let's say, a cheek cell scraping, we wanted to look at some bacteria in the mouth, then we could put a little drop of water on our slide, put our little cheek smear in that drop of water, add a little drop of stain, and then look at it that way. That allows us to increase contrast, and it's very simple, very straightforward, very quick to do. However, the method that we most commonly use is fixation. In fixation, we are going to attach the cells to the slide. And this can be done through heat or chemicals. In our lab, we will use heat. The heat also kills the specimen that we are, observing, and it preserves their integrity at that given moment in time. So what we do is we make a smear. We'll take our microscope slide. We make a very thin layer of the specimen on that surface of the slide, and that's what's called our smear, and we'll practice that in the laboratory. And then we heat it over a source. Now, in this image here, you can see we've got this warmer here, or we've got this micro incinerator or a bacticinerator. We just use the Bunsen burner or a different type of burner. Very quick, very easy to do. And what this does is it kills the cells, but it sticks them to the slide. So when we get ready to stain, we don't have to worry about our specimen, washing off the slide. So that's the first step, is making the smear. Then we add the stain. The stain is added after heat fixation. So after we fix the smear to the slide, then we add the stain. And we can use a lot of different types of stains. We have acid dyes, we have basic dyes, and we are pretty impartial when it comes to what we use. We make a positive stain, which is when we have the dye that's absorbed by our specimen, and then the background will be clear. So when we do stains, we do a positive stain. And in A and B here, these are positive stains. The background appears clear, and the specimen is what is taking up the stain. In C, this is called a negative stain. This is when we stain the background and allow the cells to show up essentially clear. Cells typically have negatively charged cell walls. So when we use basic dyes, this makes them positive stains because they're going to stick to the cell walls. We use crystal violet, we use malachite green, we use methylene blue, we use safranin, and those are our primary stains that we'll use. We don't use malachite green very often. We primarily use methylene blue and crystal violet, especially when we're doing a simple stain, which is when we use one single dye. Differential staining is when we use multiple stains in order to distinguish organisms based on their characteristics. Gram staining is the most common differential staining procedure that we use in the lab. This is, part of your lab exam, your midterm lab exam that you'll do, where you'll demonstrate the gram staining procedure. Hans Christian Gram is the one who developed this procedure in 1884, and it allows us to differentiate between the types of cell walls. So in a previous lecture, I talked about peptidoglycan within the cell walls and how gram-positive cells have a thick layer of peptidoglycan, and those gram-negative cell walls have a thin layer of peptidoglycan plus that outer membrane. The gram stain allows us to differentiate between those two. So what we do is we have a 4-step process that we use. After the after the smear is made and heat fixed, then we begin the staining process. Our primary stain is crystal violet. So we add crystal violet to the entire smear. We let it sit for 1 minute and then it's rinsed off. And so our cells are going to stain purple, kind of bluish depending on the species. But all of the cells that were in that smear will stain purple. Our next step is to add iodine. Iodine is what we refer to as a mordant, and it makes the dye a little bit less soluble. And after this point, they're all still purple. Then we add our alcohol, and we use 95% ethanol, so it's extremely flammable. And alcohol acts as the decolorizer. It washes the stain away from cells that are gram negative. So at this point, those that are going to be gram positive, they're still purple. The dye, the crystal violet, is still holding on to those cell walls, but our gram negative cells now are going to appear clear because the alcohol has washed away all of the crystal violet. The last step is to add the counterstain, which is safranin. Safranin is not going to adhere to the gram positives because they're already filled with the purple dye. They're already filled with crystal violet. What the counterstain does is it allows us to be able to visualize the gram negative cells because otherwise they would be clear. They would not show up on that clear background. So the safranin is the counterstain that's going to stain our gram negatives and make them appear pinkish or red. So gram positives will appear purple, gram negatives will appear red. And that is a differential staining process because we're differentiating our cells based on the characteristics of their cell walls. Here is a picture of what a mixed gram stain is going to look like. You've got purple cocci bacteria and you have pink rod-shaped bacteria. This is a mixed culture of Staphylococcus aureus and E. coli. Staphylococcus aureus is a gram-positive organism, and so that's why it retains that purple stain. E. coli is gram-negative, and it shows up after we add the counterstain of safranin. So if we look at this image here, we would say that this is gram positive. How do we know that this is gram positive? Because it's purple. Okay, we've had a gram stain done on this. And in fact, we could identify it a little bit more closely and say, oh, this is Staphylococcus, because we have these clusters of round-shaped bacteria. Another type of stain that we can do that's a differential stain is called an acid-fast stain, and we'll do this one in the lab as well. An acid-fast stain allows us to stain those cells that have mycolic acid. And this is a special group of bacteria called the Mycobacteriaceae, and it includes Mycobacterium tuberculosis, which is the causative agent of tuberculosis. And acid-fast bacteria tend to stain purple. They tend to stain gram-positive because they do have that thick layer of peptidoglycan in their cell wall. But it's hard to get these stains to adhere because of this outer layer of mycolic acid. You know how when you get apples from the grocery store, they feel kind of waxy? You put water on it and it just beads up on its surface. Well, that's kind of what mycolic acid is. It's this waxy lipid. And these water-based stains and dyes that we use don't really like to adhere to that mycolic acid very well. Because these water-based dyes don't like to adhere very well, we use a special process through this acid-fast stain where we use heat to drive a particular stain called carbol fusion into these mycobacterium cells. So what you're seeing here, these red cells, these are Mycobacterium tuberculosis cells. They have taken in the carbol fusion dye, and the background here has stained blue. We often use methylene blue as our counter stain for acid-fast stains. We can also do capsular staining. We're not going to do that in the lab, but it is a common lab technique where the background is stained and the bacterial cell is stained, but the capsule remains unstained. And so that's what you're seeing around these cells in this white area here. We can also do an endospore staining, where we use a differential staining where we stain the vegetative cells pink or red and then the endospores as green. We typically use malachite green for this one. Again, this is not one that we'll do in our lab, but it is a type of staining that can be done. We can also do flagellar staining that allow the flagella to show up. Flagella are so tiny and thin that we can't really get them to show up unless we use this special flagellar staining technique. Notoriously hard to do correctly, and so we won't do this one in our lab either. We mainly will stick to simple staining just to allow us to observe our specimens. We'll do gram staining and we'll do acid-fast staining.